So yeah, today I'm going to talk to you about the in situ structure of the relaxed cardiac myofibrils. And in general, we're going to see how the structure are organized both on a thick filament and thin filament perspective. So we already have quite a bit of introduction in X-ray and cardiac tissue. So I'm going to skip that and instead grab the chance to give an introduction on the technique I'm more familiar with, and that would be cryo-ET. So before going for cryo-ET, we of course have to prepare the sample, and that's where we start from. Actually, let me pause this. So our sample, as I mentioned, is the left ventricular myofibrils from mouse. We obtained this from the collaboration we have with Matthias Gautel lab. And here, what you see are what you're gonna see is actually the work from a previous PhD student in our lab, Eric. He prepared kindly this video that really showcases our pipeline. And what you see here is actually a cryo-EM grid. This is about 3 millimeters. And you're gonna see on this grid that we have the vitrified, hopefully it runs, yes. So we have the vitrified myofibrils. And of course, we obtain them by Planck freezing them. So we decrease the temperature extreme speed in order to preserve a vitrified layer of ice that is then conducive for electron and the imaging and structural preservation of the sample. Here we can see a focused ion beam view of the sample, and we use the focused ion beam to ablate away the sample, as you can see here. We can ablate away the sample, create stress relief cut. This is a top view from the scanning electron microscope. It's a dual beam microscope, so we have both the view at the same time. And our goal is to thin the sample as down in order to allow the electron to transmit and then obtain an image. So here we are thinning it down and further, and in general we aim for a 200 nanometer thickness or less. Here we have the final milling stage where, as I mentioned, we go down to 200 nanometers. And finally, we obtain these very thin lamella that are now opening really a window into our biological sample. And we can now move to the electron transmission microscope to obtain the images. So yeah, this is an image from the TEM. And here you can start appreciating although it's in grayscale, of course, you can start appreciating the myofibrils and the different structure of the sarcomere. As I mentioned, it was the work from Eric, but you can see that once we obtain this lamella, we can recognize the M band, the A band, the ZDs, and so on and so forth. And these are low magnification, so these are about 8,000x. But then what we can do is to increase the magnification, target the region of interest, and then further analyze them and obtain structural information that are higher resolution. So we can target a different region. In my case, I was obviously interested in the C zone from the A band. And then we tilt the stage in order to acquire different images at different angle. In this way, we can then reproject the images back into the Fourier space in 3D to reconstruct the final tomogram. Once we have the tomogram, the next challenge is to identify the location of the molecule. Once this is done, we can now extract subvolumes, and after the subvolumes with the molecule of interest are extracted, we can align them and finally average to obtain a higher resolution structure with increased signal to noise ratio. So yeah, as I mentioned, this was already established in our lab by Eric and Michael, and then the rest of the team, of course. And what they solved back then was the density for nebulae into the thin filament, but what they came short at was the understanding of the thick filament structure. And that's where I jump in. So for my project, in order to resolve the thick filament structure, we went for the relaxed state, because of course it's going to increase the helical arrangement of the head and hopefully help the alignment of the molecules at the first stage. So here we see a tomogram as we're gliding through bottom to top. And now, as we go back into the tomogram, we can project back the structure we solved. And here you can observe the thick filament, of course, going from the M band, T zone, and the thin filament projected back into the tomogram. Of course, the most clear identifiable features are the end-band proteins, and then we start having the myosin head in the interactive head motifs, as we did care for relaxing the tomogram with EGTA, ATP, and abundant novel content concentration. So here we have a tomographic slide with the thickened thin filament. As you can see, the raw data, and also we can observe that the heads are indeed in the interactive head motifs. Having this, I should actually point out that the average sarcomeric length is about 2.3 micron in this sample preparation, and what we observe here is the organization of the different heads. Of course, we have the M band, the P zone, and then the C zone, and the various proteins that compose it. So before talking about the thick filament, I will briefly mention what we found in terms of thin filament organization. And this is the structure from thin filament we think from our tomogram, is the calcium-free state. And what we can observe here is that we do have the block state structure in our data. Although we do observe the presence of the myosin binding protein C tethering the thin filament, we have to point out that this has been done in the physiological concentration. So of course, the stoichiometric is what it is. And also the phosphorylation state is the physiological one, so about 40% phosphorylated. As for the thick filament, so now we can have a better overview of the structure. What we solved is from M band to C zone. The D zone, unfortunately, the D zone was unfortunately unsolvable in our hands so far. And what we can see is that when we look at the projection of this structure, we can plot the intensity, and of course, we start observing the emergence of the quasi-helical arrangement with a repeat of 43 nanometers. Quasi-helical indeed, because as we can observe in the distant part of the C zone, this is more conserved, but as we get closer to the P zone and to the M band, there are quite some disturbances and helical perturbation. Before moving on, I'll just briefly give a reminder of typing organization. So of course, we have the M region, AM transition, and then we have in the A band, we have the super repeat. So we have the C type super repeat, 11 of them, and the D type super repeat, 6 of them. If we focus on into the C type, which is the part we're going to talk about today, we observe the presence of a repetitive pattern of 3, 4, 4, and with alternating fibronectin-like domain and IG-like domain. Notably, in the AM transition, when this pattern is interrupted, we have the presence of the tyrosine kinase, the pseudokinase domain, and then we have 10 domain, IG domain that go in through the M band. So with this in mind, I'm going to start the description of the thick filament from the more repetitive C zone. So in the C zone, being more orderly, we could average more molecules together and therefore further increase the signal-to-noise ratio. And here what you can see is the final reconstruction, about 18 Armstrong. This is very similar, by the way, I want to mention to the work that of course was was published in Craig and Pardon Lab by Dutta de Bradata. And we mostly come to the same conclusion as I'm gonna illustrate here. So what we can see is the presence of 2 titan chains for each asymmetrical unit. The molecule per se has a 3C symmetry, meaning that in total we do have 6 titan chains, at least in the C zone. And also, as we analyze the center of the filament, we see that we could assign the density for all the tails and justify all densities present in our reconstruction. The most prominent features here are the head in the interactive head motif. And once we assign the head, the tail, and the density for the titin, what remains is actually this additional density here in yellow. And this was assigned to the myosin binding protein C terminal domain, which I'm going to describe a little more later. I want to first go into the organization of the tails. And the third organization of the tail is a repeat of 1, 2, 3 in terms of nomenclature heads, and then we have again 1, 2, 3. So we call obviously the crowns, and we call crown 1, crown 2, and crown 3. As we individually look at them, what we can see is that they have different arrangement and probably different role in the fourth generation. So the very core of the thick filament is mainly composed by crown 3 tails. These are running mostly linearly, And indeed, if we calculate a sinusoidal compression score, we see that the compression is only 2.53 for the crown 3, while the a little more undulate is the crown 1 tail. And finally, the most undulating of all is crown 2 with a score of 4.44. In terms of radial arrangement, we have the, as I mentioned, crown 3 is the very core, and then crown 1 is an outer layer of this onion, and then crown 2 is the outermost layer present in the structure. So the difference we observe is not only the sinusoidal score, but also the interaction that they have with the different tightening chains. Here we want to break down the different interaction. Crown 3, as I mentioned, is the core and has very little interaction as far as we can tell with the different myosin, tightening domain. Different story goes for crown 1. Here we find extensive interaction of the tail, annotated here with both the tightening alpha and tightening beta. I forgot to mention this, but as we had two different tightening chains, we named them alpha and beta for a reason I'm going to explain in a couple of slides. The final tail is crown 2, and this one mostly interacts with tightening beta super repeats. And on top of that, here we also find the interaction with myosin binding protein C-terminal domain. In the reconstruction I show you, we could identify the domain from C10 to C7. However, the remaining domain and the bridging part and also the N-terminus domain was not resolved in our reconstruction. So we start assuming that this was mostly likely due to flexibility of the molecule. So to confirm this and to further investigate this connection, we went back to our raw tomogram. And here you can see a slide from a tomogram, and you can see density associated to the thick filament, the heads in lighter blue, and then the thin filament, and in yellow, the myosin binding protein C in this segmentation. So we can go back to the tomogram. In this case, what I've done was to, what we did was to reproject back the molecule we already solved into our tomogram. So we actually have the correct coordinate and also the 3D reconstruction. What was missing was the tether, and that had to be manually segmented. In this segmentation, we can observe that the tether is indeed flexible, and they do go in the direction of the thin filament, although with higher flexibility than basically merge into the density for the thin filament without clear resolution for it. Here we present the similar result where I'm just focusing on a single asymmetrical unit, or 1.5 to be more precise. And you can see how the different heads are organized and how myosin binding protein C is tethering to the thin filament, while at the same time binding to an array of myosin tails. Let's describe a little more this molecule. So we try to quantify the distribution, the angular distribution for the bridges, and we see that it's basically a Gaussian that's centered around the 90 degree and goes toward the thin filament. As for the binding of the C-terminus, what we could observe is that it's binding to an array of crown 2 tails specifically. And we have C10 and C8 that are stabilizing the interactive motif for crown 3 and crown 1. The binding of the C-terminus is better depicted here, where you can follow a single crown 2 tail. And as you go along it, you see that there are different epitopes and how the epitope is, the final epitope for the binding of the molecule is generated in the overall architecture. We want to see better how this flexibility, what the flexibility of the cross-bridge has to tell us. So to do that, we went back to the reconstruction, and this is a top view of the reconstruction. So here we have the titin and the myosin binding protein C, and then the different heads. And what you see here is the average position for the different thin filaments. Sorry, I have this in my way. Yeah, for the different thin filaments. So this is a depiction. The radial distance from the center of actin from the center of the thick filament is about 230 Armstrong. And if we average all of them together and we look at a contour map, you can observe the protrusion of myosin binding protein C, but it doesn't go in a clear direction if we average all the stripes together. If we look at the stripes independently, we have a better understanding. And it's some pattern seems to emerge. So if we look at the first stripe, we can see that it appears to have a preferential binding towards the clockwise actin, while stripe 2 seems to have a preferential binding to clockwise actin. The others seem to average out without a clear indication for a preferential thin filament. This slide is also worth noting the difference in the organization of stripe 2, which I'm going to describe later. But you can see that the heads around here are similar arrangement with the exception of stripe 2. So how does this stabilization occur? Looking at the full structure, we can focus on the first myosin binding protein C stripe. And again, just for reference and to digest this nomenclature a little more, we have named the crown counting from the first one, so P1, P2, P3, and then we start again 1 to 3, 1 to 3 for the entire of the C zone. But then we also have more precise indication of the individual crown as A1, A2, A3, A4, and so on and so forth. So if we look at the first crown 1, where the myosin binding protein C stripe 1 is located, what we can see is that there is this stabilization of the free head of crown 1 by the C8 domain of myosin binding protein C. We can dissect a little more this interaction, and we observe that it's mostly an electrostatical interaction with charges, complementary charges on C8 and the free head, as well as the C9, C8 epitope with the tail of crown 2, as earlier mentioned. So we can see that C8 stabilizes crown 1, C10 stabilizes crown 3, and C2 doesn't appear to be stabilized in any way by myosin binding protein C. However, we do observe an interaction of the free head of crown 2 with titin. And although it appears to be less strongly electrostatically charged, we do observe the contact between the titin chain and the free head of crown 2. I earlier mentioned the particularity of stripe 2, and here I want to go a little more into its quiddity. And what you can see is that to a typical myosin binding protein C stripe, so from 1 to 3 and all the others, we observe the stabilization I just described. A notable exception is stripe 2. In this case, we observe that the crown 2 is not binding to C8 from myosin binding protein C, and instead is flipping out and is going towards the actin, and its stabilization seems to be more reliant on the interaction with the light chains from crown 3, as well as its own light chain with crown 2, possibly pointing to a more direct activation or control by the beta adrenergic system and the phosphorylation in general. So now let's talk in general about the organization of these. And as we could solve the structure for the first 31 layer of myosin, we really create a blueprint for this part of the thick filament. So here we see the structure and then different models for it. And here we have an unrolled version of the cylinder to better understand the organization, in this case of the heads with their nomenclature, and then for the titin and the myosin binding protein C. I want to mention that the initial register for myosin binding protein C and for titin was found by alpha model prediction. So after the prediction from alpha model, we could very easily identify and fit this density, this model into the density, as they showed unique feature that we could identify. But even though after initially following the register from AlphaFold, we just kept following the density, and eventually ended up with the TK domain in our hand, and the only leftover density to assign it for was actually a TK M1 bulky density that really fitted the expected model. Yeah, here is a zoom in to better see it. And to describe the organization and the positioning of the head and their peculiarity, we basically decided to look at the Euler angle distribution. So we just took the central point of this triangle here and then calculate alpha, beta, and gamma angles. And as we plot them into a three-way plot, we can observe that they do cluster. Crown 2 tails are clustering together, and crown 3 and crown 1. But then we do have outliers, Some of them I described already. This is the stripe 2 I showed you before that is not stabilized by C8. There would be plenty of time to discuss all of them in other cases, in other instances, but now I want to focus mainly on the one from A1, so this guy here, and then P1, so the first one of the P zone. So here what we see is the M-band organization, and we have P1 and P2, and these were actually details that we could follow along in the density. The density for the M-band is lower in resolution as we have a limited amount of particle for this structure. Nevertheless, we could follow the tail all along, and we could see that the end of the tail roughly ends at the beginning of P1 in the antiparallel sense. On top of that, we observed the 6 transverse lines already described from previous EM studies, and we cannot ascertain for sure the identities of these, but we can already identify some M-band proteins that are, of course, coming out from the density, and then they average out as they are flexible in nature. In terms of typing, as I mentioned before, we could easily follow the different domains down to A170 and then the presence of the TK domain and M1. Then from previous study, we know that the structure encompasses the M-band going to the other direction, and here we are proposing a model of how this might going. This model is based on AlphaFold prediction and the calculation for the interspacing sequences we have. But of course, the resolution at the moment it's limited, therefore we cannot make final assessment on the nature of these densities. But let's talk about the P zone. So as I mentioned here, you can see the alpha typing chain, and that's the reason why we call it alpha, because it goes all the other way. And we can observe that getting to the P1 crown, we see a bulkier density that really resembles the TK and the M1 domain, while the M2 and the M3 are wrapping around the tail for crown 1. The absence of the other chain is then explained by this model. If we focus in this region, if we focus in this region instead, what we could observe was the presence of additional density that was previously reported as a possible accessory protein that was never actually identified in the thick filament. And what we're suggesting is that what happens is that the typing beta chain, after arriving to the crown 2A2, it forms this bow tie structure holding to itself, and then finally ends with a TK M1 domain onto the crown 1A1 head. I want to show you this brief animation. This is the actual model, the raw data, and here I'm fitting in the PDB models for all the density. And now I'm just exaggerating the structure to better illustrate the organization of alpha and beta. So here we have the alpha typing that goes all the way to M3 on top, sitting on top of P1. And then if you go below in the structure towards the M-band, so the Z line, we observe this density. Here is the density we could not assign, and these are just 3 Ig domains that are most likely flexible and not solved in our density. This is the raw data, and you can really see how the fitting for the TK and M1 domain seems appropriate in this case. So with this, I'm gonna jump to the conclusion. What I showed you today was the relaxed cardiac myofibrils. We went from the thin filament to the thick filament, and what we could observe is the general organization of typing. And with this organization, we can start making some assumption and some proposal as to how this structure is regulated. Especially if we focus in the better resolved T zone, we can see how this structure can really sense, integrate, and then modulate the response to multiple signals. So the kink nature of the typing chain really suggests a possibility for strain sensing and for the relaying of the signal to the myosin tail, and in particular to the crown 2 head. So in this model, crown 2 actually acts as a sentinel head that can then relay the signal to the other components. And as it does this function, we can imagine that crown 2 heads, the first to be activated, the most undulated one, and also the one that generates the array for myosin binding protein C to Phyton, can then lead to the destabilization of this epitope and the destabilization of the binding for C8, and then the interactive head motif of the other crown, in particular crown 3 and crown 1 later on. So with this in mind, I want to thank all the people involved in this project, in particular Matthias Gattel lab for providing the sample, Pauline for the microscopy data, Aileen for the sample again, and Martin for the antibody we used in the super resolution microscopy that I haven't had time to show here. And from Rounds and Lab, I'd really like to thank Michael, Marcus, Sebastian, and Thorsten for their help in the preparation of the sample, and specifically Thorsten for scripting, Eric for teaching me most of what
we couldn't hear you.
Thank you for your attention.
Okay.
Yeah.
Last part. Okay, we have two questions in the, a couple of questions in the chat. You want to take it, or you can just unmute yourself, ask it directly, ask the speaker.
If they can do that, wait, I think I have them. Let's go from the first one. How did you get 2.3 in cardiac unloaded myofibrils? This was just what we get. So in terms of sample preparation, I might relay this to our collaborator, but what we did was just to have them. Sorry, I hear multiple. Yeah. So the sample was prepared in Rigor state, and then all I did was to add the ATP, add the mavacamten first, and then the ATP last, just prior to Planck freezing. Does that answer the question? I'll assume so for the moment, but if
I have a question. Yes. You know, well,
yes,
I look your images, and especially cross section of backbone. So, how many tails you have in cross section? LLMs.
Yep.
I account
30. I believe that 30. It does depend where you cut, of course, which section you're looking at. It might be
well, but
I believe 31
if
or
you
36.
If you look in C zone, for example, in C zone, how many you have in backbone?
They are all the need to be according to the calculation I made, but I don't have the number now in mind. What would
I look there are some pictures.
Yeah, let's go.
In cross section.
Yeah. I'm going there. Let me go there. Yeah.
Oh, like this. I look these. So if I look ground 3, okay, I presume these are tails. These round things, because they go, you know, as a circle because of helix 1. So it's hard to see what is in backbone. I see in center 6. Okay, and after after that they are a little bit kind of distributed in certain way, and maybe we can talk directly about these details, not to bother others, because another thing is I like to see how force is transmitted through backbone, and there is some distance between these, how many of them. I account technically just there should be about 18, but how you put and also we have S2s which are now very close to it, so it's hard to distinguish which is S2 and which is that backbone part. So I calculate roughly about 18. I don't know what you got, but
they should be. Well, so one thing to consider is that do not actually none of them circles around the structure. They all run fairly